This is going to complete the overall lecture topic of metabolism, finishing the second part on biochemistry. We're going to talk about electron transport chains and oxidative phosphorylation, which is also a discussion of the process of chemiosmosis, which means using a proton motive force to generate ATP. What I'd like you to learn from this lecture and the accompanying reading in Chapter 4 is that I would like you to have an understanding of how electron transport chains are constructed and organized in the cell. I'd like you to understand the flow of electrons from electron carriers through electron transport chains, which I will be abbreviating ETC, okay, and ultimately to terminal electron acceptors. And I'd like you to understand how the proton motive force is generated, what a proton motive force is, and how it energizes ATP synthase. So respiration can be aerobic or anaerobic, and if it's anaerobic, then you're using a terminal electron acceptor other than oxygen. Missing a closing bracket there. The terminal electron acceptor in respiration is always exogenous or external to the process. Okay. The goal in respiration is to transfer electrons from the primary donor to the terminal electron acceptor. At the level of the electron transport chain, the electrons are moving from electron carriers, such as NADH, to the terminal electron acceptor. So during glycolysis and the Krebs cycle, we've stripped electrons off the original primary donor and its subsequent metabolites. We've placed all those electrons onto electron carriers, such as NADH, or also FADH2. Or if, for example, in photosynthesis, it could be NADPH, okay? And then those diffusible electron carriers have diffused to the protein complexes that make up the electron transport chain. At the electron transport chain, we have those electrons flowing through the components of the electron transport chain and ultimately to the terminal electron acceptor. The electron flow is energy, and that energy is harnessed to create a proton motive force, and that proton motive force is then harnessed to run ATP synthase and also to accomplish other important functions such as transport or running a flagellum. Electron transport chains are membrane-associated suites of electron carriers, including enzymes and various molecules. Remember that this is all about redox. Electrons are always transferred in an electron transport chain from one chemical species to another, and it always has to be from one chemical species to another that has a more a more positive redox potential. So let's look at electron transport chains in some detail. So let's think about what's in an electron transport chain. Electron transport chains are always found in membranes, either the inner mitochondrial membrane in a eukaryotic organism or the cytoplasmic membrane in a bacterium or archaeon. They include complexes of proteins. Here we see complex 1, 2, 3, and 4 in brown. Those complexes include multiple proteins, and the kinds of proteins found there include dehydrogenases, flavoproteins, iron-sulfur proteins, and cytochromes. I will talk about those in a moment. Also found in an electron transport chain are quinones. That's what's shown as Q. Sometimes that's referred to as coenzyme Q or CoQ. They are small organic molecules that I talked about in a previous lecture. There's also an important protein not typically found in a complex, but that is part of most, or actually all, electron transport chains, and that's the cytochrome C protein. It acts on its own, and it's usually on the exterior side of the membrane. Notice that these complexes and other electron-carrying molecules are organized so that electrons are transferred always to something that has a larger, or a more positive, redox potential, or E0. So electrons will come into the electron transport chain either in the form of NADH or sometimes in the form of FADH2 or other molecules. Okay, so they're coming in here and in here. Let's just focus on complex 1 first. Complex 1 contains multiple proteins. They're going to remove electrons from the NADH and they're going to transfer them to the quinone pool. From the quinone pool, they're going to be transferred to complex 3. From complex 3, they're going to go to cytochrome C. From cytochrome C, they're going to go to complex 4. And from complex 4, they're going to ultimately end up on the terminal electron acceptor. In this case, it's oxygen. And that's the end point. Water is formed, and that's the end of the road for these electrons. Electrons that come in as part of FADH2, for example, from the Krebs cycle, enter at complex 2. They go from complex 2 to the quinone cycle, from the quinone cycle to complex 3 to cytochrome C to complex 4, and from there again to the terminal electron accepting molecule. Let's think a little bit about these different proteins and components more. Some of that should be reviewed from an earlier lecture. Complex 1, which receives NADH, includes several proteins. I think it's 14 or 15 in E. coli, for example. Those proteins include a really key protein called NADH dehydrogenase. Okay? And that NADH dehydrogenase is what actually binds to NADH and accepts electrons and protons from it. It accepts 2 protons and 2 electrons, and that forms NAD plus and H plus. That's going to go back to glycolysis or the Krebs cycle or whatever. This is going to actually participate in the activities of generating the proton motive force. The electrons are going to go from NADH dehydrogenase to a flavoprotein. Flavoproteins contain molecules like FMN, and then they're going to be transmitted to an iron sulfur protein. Iron sulfur proteins were discussed in the previous lecture. And then ultimately they're going to end up in the quinone pool. It's all of these together that form the green blob that is called complex 1. I mentioned in an earlier part of this lecture what a flavoprotein was. Flavoproteins are proteins that have prosthetic groups that are either flavin mononucleotide, FMN, or flavin adenine dinucleotide, FAD. Both of these prosthetic groups, either FMN or FAD, are derived from riboflavin, which is an essential vitamin for many different organisms. These are prosthetic groups that can accept protons and electrons but transfer only electrons. And the flavoproteins that carry FMN are found in complex 1 of most electron transport chains, whereas flavoproteins that contain FAD are found in complex 2 of most electron transfer chains. You've looked at these previous, but here we see oxidized and reduced forms. You can see where the reduction has resulted in the FADH2 form here, and these are really important electron carrying molecules that are associated with proteins called flavoproteins found in electron transport chains. What else is in electron transport chains? Well, really important components of electron transport chains are cytochromes. So cytochromes are proteins that contain heme groups, and a heme group is shown in here, okay, associated with the brown, which is protein. Okay. So hemes are prosthetic groups, right? They are part of proteins so that they can accept and transmit electrons. So hemes are capable, or most cytochromes in fact, which contain hemes, are capable of 1 electron transfers, and that is mediated using the iron, the Fe, in the center of the heme group, shown here. Okay. There are a lot of different kinds of cytochromes in cells. So while they've been evolutionarily conserved into classes that are designated by letters, as for example cytochrome A, cytochrome B, cytochrome C, there's still some diversity within those classes, and so there's an additional numbering system. For example, you can have cytochrome A1 or cytochrome A2 and so on. Each species will have its own particular suite of cytochrome proteins. These proteins are really good at complexing with other cytochromes and also complexing with iron sulfur proteins, which we'll talk about in a moment. They don't carry or transfer protons, so they just accept electrons and they just transmit electrons. They themselves don't sort of manage or deal with protons. In electron transport chains, they are found associated with complexes 3 and 4. Electron transport chains contain iron-sulfur proteins, which contain iron not in the heme form, but instead complexed with sulfur. Those prosthetic groups are typically associated with the protein shown here in brown with the help of the amino acid cysteine. Cysteine is very important in coordinating the location and attachment of these prosthetic groups. And these iron-sulfur proteins play a very major role in complexes 1 and 3 of most electron transport chains. They carry electrons, but not protons. Quinones, also known as coenzyme Q, are shown here. They're very important small organic molecules. They're very hydrophobic. They're found associated with the fatty acid tails in the membranes that contain electron transport chains. They are somewhat mobile within those fatty acid tail areas of those membranes, and they accept protons and electrons but transfer only electrons. These are really essential molecules because they connect complexes 1, 2, and 3 together. Let's now put the different parts of an electron transport chain together and think about electron flow and think about proton production and pumping. First, let's address the protons. Here we've got protons, and they are accumulating to create a proton motive force. This is the outside, this is the inside of the cell, okay? Over here we've got ATP production. Remember, ATP is always produced inside the cell. Cells never make ATP on the outside because that would be wasteful. So that's one way that you can always tell what's the inside and what's the outside. All right, the protons, where do they come from? They come from two places. One, they come from NADH. Okay, when it's oxidized, it yields protons. And two, they come from water dissociation. Okay, so that's the sources of the protons, and they are being actively pumped using these proteins embedded in the membrane. Is altogether called the electron transport chain. They're being actively pumped so that they accumulate on the outside of the cell, creating an electrochemical gradient known as the proton motive force. All right, let's think about the electron flow now, because it is the electron flow that provides the energy for these protein complexes to pump protons across the membrane. So, this is a simplified version of an electron transport chain than the one shown in your book. And what is not shown here is the complex that your book refers to as complex 2, which would be at about this position, and in the figure in your book, which we'll look at next, plays the role of accepting FADH2. This is a simpler version as is found in other cells. Okay, there are different versions of electron transport chains in all the different critters on Earth. So here we have NADH coming from other places in the cell like glycolysis in the Krebs cycle, and it is attaching to this protein, and this protein is often called complex 1. It includes NADH dehydrogenase, which catalyzes this oxidation. That starts to get the electrons flowing at the electron transport chain. So electrons are coming from NADH into the NADH dehydrogenase, and this may also have iron-sulfur proteins in it and flavoproteins in it. Okay, the electrons will flow from one to the other, always from something that is a very negative redox potential to something that has a more positive redox potential, and then ultimately they'll end up in the quinone pool, which is shown here. So here quinones are being reduced and then oxidized as they drop electrons off at the cytochrome BC1 complex, which is complex 3. This was 1. For comparison to the figure in your book, 2 is not shown, but it would be down here. Okay, so cytochrome BC1 complex contains cytochromes of B type and C1 type, and they're going to transmit electrons one to the other, and then they're going to transmit them to the protein known as cytochrome C, which is actually attached to this membrane, not loosely floating in space as this figure implies. The electrons will go from cytochrome C over to complex 4. And at complex 4, they sort of meet the end of the game from the point of view of electrons, in that they are going to end up on oxygen, reducing that oxygen to water. Okay, and so there is the terminal electron acceptor. Okay, that's the end of the game for the electrons. That electron flow is what's providing the energy for the active extrusion or pumping of protons, creating the proton motive force, an electrical potential. And that electrical potential is harnessed by the enzyme ATP synthase, so it has a channel through which protons flow moving down gradient back into the cell. That energizes ATP synthase, allowing a rotor to turn, and that allows ATP synthase to catalyze the reaction whereby ADP and phosphate, inorganic phosphate, join to form ATP. All right, let's look at the figure in your book that puts all this together. It's more complex, so I wanted to look at this simpler figure first. So let's look at a figure that is similar to what's in your book, although it is oriented kind of sideways, okay? And I think that makes it easier when thinking about the redox potential. Here is an electron transport chain as seen in Paracoccus denitrificans, as I said before, a well-studied organism in this regard, and it includes complex 1, 2, 3, and 4. Let's track the electron flow. Electrons come in on NADH, and that is generated from either glycolysis or the Krebs cycle. And the electrons on NADH are transferred to complex 1, which contains NADH dehydrogenase, not shown but here, okay? It contains flavoproteins, which hold FMN as electron-carrying molecules, and it holds iron sulfur proteins. The electrons are going to flow from NADH through complex 1, okay, and into the Quinones cycle. Note that protons are pumped through as that happens, and the energy for moving the protons through comes from transferring electrons sequentially to more positive, to substances with more positive redox potential, E0. The electrons will enter the Quinones cycle, and they will be then transmitted to complex 3. Okay? As part of the activities of the Quinones cycles, more protons are pumped to the exterior of the membrane. Note that the Quinones cycle can also accept electrons that come in from complex 2, and those would be electrons that are coming from FADH2. FADH2 is generated by the actions of the Krebs cycle. This is also a place where electrons that enter in the form of succinate in some organisms can be accepted. Those electrons will flow to the Quinones cycle, and then they will flow to complex 3. And as that happens, protons are pumped. Complex 3 contains cytochromes, cytochrome B proteins. It contains iron sulfur proteins, and it transmits electrons sequentially from cytochrome Bs to iron sulfur proteins, and then off to cytochrome C, which is typically associated with the exterior portion of the cytoplasmic membrane in microbial cells with electron transport chains. The electrons are then transmitted from cytochrome C to complex 4, which contains cytochrome A proteins. And from there, the electrons are transmitted to the terminal electron acceptor. In this figure, it is oxygen. So this is an aerobic respiratory organism. However, if this was an organism that was capable of doing anaerobic metabolism, instead that oxygen would be something like nitrate or sulfate or some other electron-accepting molecule. Note that the activities of complex 4 result in protons being pumped to the exterior part of the membrane. So overall, the energy obtained by these sequential redox reactions is utilized by complexes 1 and 4, as well as the Quinones cycle, to pump protons to the exterior part of the membrane, creating the proton motive force, an accumulation of protons on one side of the membrane in which the electron transport chain is found. Also note that electrons are always being transmitted from molecules that have a more negative redox potential to molecules that have a progressively more positive redox potential. So here we have plus 0.39, up here we had minus 0.22. Electrons are always transmitted in this direction, which would be downwards in the electron tower that we looked at in previous lectures. And finally we get to ATP synthase and the proton motive force. So ATP synthase is a multi-protein cytoplasmic protein complex that is embedded in the cytoplasmic membrane. It's a complex organized into the F1 region, which is oriented on the inside or the cytoplasm side. So that's in here. Sorry, this is the opposite of the figure that we looked at 2 slides ago, and this is out. Okay? Remember, ATP is always produced inside the cell. So let's look at the different bits and pieces here. Here's the F1 part. It's a protruding knob that contains catalytic sites that join inorganic phosphate to make ADP in order to make ATP. So that's its job. It's to actually catalyze this reaction. That's the F1 portion or region. We also have a rod that connects the cylindrical rotor portion, which we're going to get to next, and spins it, activating catalytic sites in the F1. We have the F0 or F0 site, which functions like a motorboat rotor, and it is found in the cytoplasmic membrane. It spins clockwise when it's doing the work of ATP synthesis. So when protons are flowing through it down the proton gradient, when they're flowing this way, okay, then it will spin clockwise like this, and that will cause the rod to spin, and that will trigger catalysis due to the F1 portions. So this is a very ancient protein. It's been highly conserved. All cells have ATP synthase, and their job is to make ATP. They're also used for some other processes. So this is oxidative phosphorylation, which is the synthesis of ATP utilizing ATP synthase, which is energized through the proton motive force. I want you to also note that ATP synthase in phototrophic organism works in the same way. It's just that the proton motive force is generated using energy from photosynthetic reactions instead of the successive oxidation of high energy compounds. So in that case, we call it photophosphorylation, something I mentioned in an earlier lecture. All right. So using the resources that you have available to you, chiefly Chapter 4 of your textbook, I want you to use and answer this study question. Fermentative microbes still have ATP synthase. What for? And a hint as you go about answering that question is that in addition to running ATP synthase and using it to generate ATP, what is the proton motive force used for in cells? In other words, why would a fermentative microbe, just like a respiratory microbe, need to have a proton motive force? You should be able to list in total 3 purposes for a proton motive force, and that should give you an indication of why fermentative microbes still have ATP synthase. Another hint that I haven't written here, but it is that in some organisms ATP synthase can actually run backwards. So think about that a little bit, and please comprise an answer to this question for your study notes. With that, this will now conclude this last lecture on the topic of metabolism.